Both dendrites and axons are extensions of the membrane that surrounds the nerve cell body of a neuron. Dendrites are the structures that will be receiving information from other neurons. Dendrites can number between 1 and thousands per cell. There are neurons that have a single dendrite associated with them, but many neurons have branching levels of dendrites. One dendrite branches into 2, and each of those branch into 2, and so on, until there are thousands of individual dendrites available to receive sensory information. The axon also differs in number. While many neurons have a single axon, there are neurons that have an axon that branches somewhere along its length. That creates what can be thought of as a primary axon, and then what is called a collateral or a branching axon. The junction between the axon and the nerve cell body of a neuron is a specialized region referred to as the axon hillock. We'll talk about the role of the axon hillock when we talk about how electrical signals are passed from neuron to neuron. Down at the end of the axon, the most distal end of the axon, there's also branching evident. The axon will branch out, and then each branch will branch again into what are referred to as terminal branches. Each one of the terminal branches ends in what's called a synaptic knob, or a synaptic swelling, or a terminal button, or a terminal knob. These enlarged regions of the terminal branches go by lots of different names. Axons that come off of a neuron can be myelinated or non-myelinated, depending on where you are in the nervous system. Myelin is a fatty material that gets secreted by certain types of cells within the nervous system. Myelin can be thought of as a layer of insulation around the axon, and just like the insulation that you would find around an electrical wire, myelin allows for electrical impulses to travel down an axon without any loss of signal. Now, as I said a moment ago, there are neurons that have as few as one dendrite. There are some neurons that don't have an axon, but most of them do. Slide, we're looking at drawings of axons and dendrites on individual neurons, and we're looking at an image that was taken underneath an electron microscope. Underneath the electron microscope, you can clearly see the cell body of a neuron, and then you can see all of these membrane extensions coming off of that cell body. These make up the dendrites. One of them is likely an axon, perhaps this one. All of these structures that come off of the cell body are simply extensions of the neuron. So into each dendrite, into the axon, if that's what this one is, there is cytoplasm in there. It's an extension of the cell, and it's wrapped in cell membrane. Over here in yellow, you can see a myelinated neuron. Here's the cell body with the nucleus and the other organelles. These are the branched dendrites extending off of the cell body. Here's the myelinated axon right here. You can see little white sort of cotton ball structures here along the length of the axon. This is the myelin. In between each one of these white structures, these white sort of mounds of myelin, there is an area that doesn't have any myelin on it. So the myelin sheath is not continuous. It's disrupted every so often at areas that are called nodes, and more specifically, nodes of Ranvier. So the myelin sheath is not continuous. It has these areas every so often that have no myelin around them. Down at the bottom of the axon, we can see a branching pattern. The final branches that are created are referred to as terminal branches. Down here on the bottom of the slide, we can see a drawing of one neuron here. It happens to be myelinated, and it is communicating with two other neurons. So here's the nerve cell body in blue, and the dendrites coming off of it. These dendrites are receiving information from some other cell or some other sensory receptor. And this cell body is passing the information along the length of its myelinated axon. You can see the branching pattern down at the end of the axon, and some of these branches are communicating with this cell. Some of these branches are communicating with this cell. The ends of the axon, the terminal branches of this first cell, are going to send along the electrical signal to these second and third cells from the axon to the dendrites of the next cell. And then that signal will travel down each of their axons and be passed on to the next cells. Now we're going to spend a few minutes talking about different types of neurons and other cells that are found in the nervous system. But the first thing I want to talk about is just some very general types of neurons. When we talk about neurons, one of the ways that we can distinguish them is based on how the dendrites and the cell body and the axon are oriented in terms of each other. So far we've been talking about a cell that looks like this. This cell, you can see its cell body right here with the nucleus dark in the middle. You can see dendrites coming off of the cell body. You can see a single axon, happens to be myelinated, and then you can see terminal branches and terminal or synaptic knobs at the end. This is the cell we've been looking at so far. This is what we would call a multipolar neuron. Now I want you to look at the cell that's sitting next to it. This cell looks very different from what we've been talking about so far. Instead of seeing dendrites emerging right off of the periphery of the cell body, instead we see a single membrane extension that then branches into dendrites. And again, on the other side, we can see an axon, terminal branches, and then the knobs again. This is the kind of configuration that we refer to as a bipolar neuron. Two poles or two distinct membrane extensions. One of them is going to become the dendrites, and one of them serves as the axon. Now, if you go over all the way to the right, you see a cell that looks even more unique. Here's the cell body of the neuron, and this time there's only one extension coming off. This is what we call a unipolar neuron. Now it's still going to have dendrites and an axon. They're just ordered differently here. From the single extension, we'll see a branching event right here that creates the dendrites. And then from the dendrites, there'll be a single branch emerging that becomes the terminal branches of the axon. So again, a single membrane extension, that's why we call it unipolar. And that single extension first branches into dendrites, and then creates the axon and terminal branches. Now this one in between, this is called a pseudo unipolar neuron. It's technically unipolar because there's only one membrane extension coming off of it. But as you can see in the picture, it very quickly sends a membrane extension this way, which branches into dendrites, and a membrane extension this way, which is the axon. So it is unipolar, but it's not like this cell is because of the way it branches. So unipolar, one membrane extension; bipolar, two membrane extensions; pseudo-unipolar, one membrane extension that quickly becomes two; and then the most traditional multipolar neuron. Now, how common each of these types of nerve cell configurations are depends upon where you are in the nervous system. Depends upon whether you're in the central nervous system or the peripheral nervous system, and where you are in each one of those. So now that we've looked at the different shapes that neurons can take, let's talk about the different functions that neurons and other cells of the nervous system perform. When we talk about nervous tissue, there are two primary cell types. Remember, all tissue contains cells and extracellular matrix, and nervous tissue is no different. So when it comes to cells, there are both neurons and there are what we call glial cells. Now this word glial, when you translate it, it means glue. Back in the early days when scientists were first investigating the anatomy and the physiology of the nervous system, they actually believed that glial cells were there to simply hold together all of the neurons inside the nervous system. So they honestly thought that these glial cells didn't do anything else except occupy space and hold everything else together. We now realize that glial cells perform a number of different functions. And we'll talk about each of the types in just a moment. But when it comes to the neurons in the nervous system, there are three types. There are what we call sensory neurons, there are what we call motor neurons, and there's a type that we haven't mentioned yet called interneurons. For glial cells, which are found both in the central nervous system and the peripheral nervous system, there are microglial cells, there are astrocytes, there are oligodendrocytes, and there are Schwann cells. Let's begin with the neurons. Sensory neurons, as we've discussed so far, are the neurons that are receiving information from the environment or from the internal parts of the body and are relaying it to the central nervous system. So they are providing information from the periphery, from either the outside world or from regions within the body, and they are sending it along afferent pathways to either the brain or the spinal cord. I wrote brain here, but it should be brain and spinal cord, the central nervous system. When we think about sensory information, we often think about things like sight and smell and touch, but remember, sensory information also includes things like pressure and stretch and pain and temperature, any sort of information that can be perceived by the nervous system. So sensory neurons carry information toward the central nervous system along afferent pathways. The central nervous system then integrates all of that information and responds. The response is sent along motor neurons in efferent pathways. Motor neurons are providing the response. So motor neurons can provide movement information that travels from, again, the central nervous system, brain and spinal cord, to the skeletal muscles. But motor information can also be sent to glandular tissue, to both exocrine and endocrine glands. These are efferent pathways. These are response pathways. Remember, for both sensory neurons and motor neurons, we're talking about some of them being somatic or voluntary and some being autonomic or involuntary. And along the involuntary pathways, some are sympathetic and some are parasympathetic. So they're all motor neurons, but they can fall into different types. Same thing with the sensory neurons. Finally, there are interneurons. Interneurons actually make up 90% of all of the neurons in the nervous system. That's an amazing number. Interneurons function to receive information from one neuron and simply transmit it to another neuron. So they are the middlemen neurons. They receive information from one and send it along to another. Now, one job that interneurons have that's very important is this. When they receive information, they sometimes receive it from a single cell, but other times they're receiving it from many other neurons. And so they have to be able to integrate that information. If they're receiving information from one neuron, they simply have to pass it on to another. But if they're receiving information from 2 or 3 or 10 neurons, they have to then integrate it so it can be passed on to the next neuron. Now let's take a minute and talk about glial cells. Glial cells are the most abundant cell that you find in the central nervous system. That surprises people when you tell them that, because you would imagine that neurons are the primary cells in the central nervous system. But it's the glial cells, the supportive cells, that make up most of that tissue. Remember, that term glia means glue or support. We now know, again, that there are 4 types or subtypes of glia. There are microglial cells, astrocytes, oligodendrocytes, and Schwann cells. Microglial cells are actually immune system cells that live and function in nervous tissue. These are phagocytic cells, so when they encounter either foreign material or infectious material like bacteria or viral particles, they go through phagocytosis and they take that foreign material inside of themselves and they destroy it. Astrocytes are probably the most interesting of the glial cells because they have many functions. We probably don't yet understand all of the functions that astrocytes perform in the nervous system. One of the most well-known functions is that astrocytes make up a structure called the blood-brain barrier. As the name suggests, this is a physical barrier that prevents things from coming out of the blood and gaining immediate access to cells in the brain. This is a protective barrier, and it is made up of astrocytes that are side by side surrounding the brain. The astrocytes carefully control what things can get into and access to the brain. Other jobs that the astrocytes perform, we know that they help modulate the signal that comes to a synapse, that comes to a point of communication between cells in the nervous system. They help impact how strong or how weak that signal is going to be. We also know that they play a role in chronic pain. Chronic pain is a problem that a lot of people suffer with. And unlike acute pain, the kind of pain that you would feel, say, if you fractured a bone or if you lacerated your arm, chronic pain is its own disease. The pain sensing cells in the body start to malfunction, and it's believed that astrocytes play a role in that very unusual process. The other two glial cells that are in nervous tissue include oligodendrocytes and Schwann cells. Now, both of these cells have the same function of secreting myelin around axons. The difference between the two cells is where they are functioning. Oligodendrocytes are producing myelin sheaths in the central nervous system, and Schwann cells are forming myelin sheaths in the peripheral nervous system. On this slide, we're looking at some drawings of individual cells that are found in nervous tissue. This cell over on the left is representative of a neuron, and if you remember from earlier, we would refer to this as a multipolar neuron. And then you can see some glial cells. 3 of the 4 glial cells are shown here. This is an astrocyte, this is a microglial cell, and this is an oligodendrocyte. So the only cell that's missing is the Schwann cell. The astrocyte looks unusual. It doesn't look quite like a neuron, but it certainly doesn't look like an ordinary type of animal cell or human cell because it has all of these membrane extensions. Remember, this is not a neuron, but this is a cell that performs many different functions within the nervous system. The microglial cell here also has membrane extensions coming off of it, but remember, this is an immune cell, and it got its name micro because it is very small. Finally, here's the oligodendrocyte. What's most interesting about this cell is that it sends out membrane extensions to a nearby axon, and that's what this is. And it is along those membrane extensions that it produces and secretes the myelin that will coat this axon and give it that insulation layer. On this slide, we're looking at Schwann cells producing a myelin sheath in the peripheral nervous system. Up at the top, this structure right here that looks like the letter C, this is a Schwann cell. And what it's doing is it's wrapping itself around an axon, shown in yellow. So if this is what an axon looks like along its length down here, what we've done is we've cut a transverse plane through the axon, and we're looking at its cut end right here. So the Schwann cell surrounds the axon physically, sort of hugs the axon. And as you can see from here to here to here, it wraps itself around not just once, but it wraps itself around so that the axon is completely surrounded. And then the cell starts to secrete layer upon layer upon layer of myelin. So when you look down here at the bottom of the slide, this is one of those cotton balls of myelin that gets laid down on top of an axon. Right here, they've cut it in half so we can peek inside and see that it's actually produced in layers. This is the area in between this sort of mound of myelin and this mound of myelin. This is that unmyelinated area called a node or node of Ranvier. Schwann cells physically wrap themselves around the axon. That's different from oligodendrocytes. Oligodendrocytes sit nearby and send out membrane extensions to secrete the myelin. Schwann cells literally sit on top of the axon and secrete myelin. On this slide, we're looking at an image that was taken underneath a light microscope of various cells that are inside nervous tissue. These happen to be motor neurons and glial cells. Now, the motor neurons are the larger cells visible on the slide and the ones that are staining more darkly. You can see inside each motor neuron a circular nucleus staining darker purple. And on some of them, you can really see the membrane extensions coming off of the cell body. These would be dendrites and an axon. So some of them, you can see the dendrites really clearly. It's a little bit hard to tell where axons are. Generally speaking, axons are the largest of the membrane extensions, but it can be hard to tell which is which on a preparation like this. Now, all of the glial cells that are visible in this tissue are much smaller than these individual motor neurons. So all of this pink material back here and all of these little dark circles, these are individual glial cells. It's hard to tell where one cell ends and the next cell begins, but all of the individual dark purple dots are nuclei. So you can see there are many, many cells out here surrounding these motor neurons. These are all different kinds of glial cells. Some of them are microglial cells. Some of them are astrocytes. Now, we're not told in this image whether we're in the peripheral nervous system or the central nervous system, because of course motor neurons can be found in both of those. But if we were in the peripheral nervous system, it's likely that there are Schwann cells that are associated with some of these motor neurons, myelinating the axons. If we were in the central nervous system, it would be oligodendrocytes that would be associated with some of these motor neurons, providing that myelin sheath. Remember, not all axons are myelinated. Only certain axons are myelinated, both in the central nervous system and the peripheral nervous system. One more thing to note about myelin. You've probably heard of the disease called multiple sclerosis. Multiple sclerosis is a disease of myelinated axons, and specifically, it's an autoimmune disease where the person's own immune system attacks and destroys myelin. Multiple sclerosis is actually a series of diseases. There are different forms of multiple sclerosis, and each form has more or less severity. It's also an unusual group of diseases because it can strike at different parts of the nervous system. For some people, it strikes out in the peripheral nervous system, out, say, in the fingers or in the toes or the legs. In other people, it starts in the eyes. It's a very unusual disease. But across all forms of multiple sclerosis, the basic problem is that the immune system is seeing myelin as a foreign material, and therefore it is destroying it. And axons that were intended to be myelinated, in other words, intended to be insulated are losing that insulation, and so they are not able to transmit the electrical signal as efficiently as they once did.